Alright, everybody, welcome back to Kember. I'm pretty excited for this video today because, as you guys may have heard from just in passing in other videos, I've recently graduated college, which, let's go, that's awesome, right? But another cool thing is that I recently defended my honors thesis, and luckily, I thought on my feet and was able to record my thesis defense. And I've talked to my principal investigator. He's happy with me posting all of this stuff online. So I guess here we are. I just want to share what I've been researching for the past couple of years online, and I guess just give people a view of what is currently being done with NIH funding. I think it's especially important in this time to be transparent about what type of research is being done. And I think my project is really cool, and I'd be honored to share with other people and what benefits may come from this research. So, to preface this, my research is in the field of structural biology, which looks at molecular interactions between various kinds of molecules. In my case is protein-protein interactions, and I guess using empirical methods to determine whether we can improve expertise in the medical community through our findings and increasing our knowledge of certain signaling systems such as ion channels, which I focused on in this honors thesis, but you'll hear more about that. I also wanted to mention that this video is going to be pretty extensive, and for people who didn't really get a chance to ever study biology past the high school level, this video might be pretty daunting because this is I guess a culmination of really tough effort of really trying to learn as much as I could about molecular biology, and it really took a lot of effort to get to the point where my project really makes sense and I can see the light at the end of the tunnel, why I'm actually doing all of this work at the end of the day. So it's really tough to do that on your own without biology degrees. So I plan to, in the future, in like a month or two, I'm still relaxing a little bit after graduating, but once I have my relaxation, Camber will be back and I'll talk about my thesis more in depth. But for now, I hope you enjoy watching this recording of my thesis defense. Yeah, I hope you enjoy. My honors thesis I'm going to present to you guys today is a structural analysis of OSC1 binding to the nicotinic acetylcholine receptor using cryo-EM. So just some background real quick on OSC1. It's an alpha neurotoxin peptide produced by this scorpion here called Orthochirus scrobiculosis. And so the way this toxin binds or acts is by binding to the nicotinic acetylcholine receptor, as well as potassium channels, which is found in Kashavarov et al. When this toxin binds your acetylcholine receptors, it causes flaccid paralysis. So to understand how it causes flaccid paralysis, we need to look at the structure of neuromuscular junction. So in this junction, we have acetylcholine receptors, which are pentameric ligand-gated ion channels. So these receptors are found peripherally as well as in the central nervous system in your brain. But in this talk, I'm going to only be mentioning the peripheral nicotinic acetylcholine receptors found in your muscles. So what happens is when your brain sends a signal that you want to move your arm, it's going to cause a release of acetylcholine from this presynaptic neuron. The acetylcholine is going to cross the synaptic cleft, bind the acetylcholine receptor, and this is going to cause the channel to open, and sodium ions are going to go into the muscle cell. So when these sodium ions go into the muscle cell, this then causes a depolarization of that cell membrane, which leads to calcium release from the sarcoplasmic reticulum. This calcium will then diffuse into the sarcoplasm and bind myosin, which is it's a motor protein which this motor protein, when it's bound with calcium, can then exert a force, causing you to use your muscles to contract. So if you want to prevent your muscle contraction, you can prevent this influx of sodium to cause flaccid paralysis. And so some drugs that are already used to promote flaccid paralysis are these neuromuscular blocking drugs. And the way they bind is that they mimic the quaternary amine of acetylcholine. So this is the neurotransmitter that normally causes muscle contraction peripherally. What you can see here is this nitrogen, which is bound to 4 groups, has a positive charge. You can see these are 2 examples of neuromuscular blocking drugs, and they both also have these quaternary amines, which they use to bind the acetylcholine receptor. So these drugs are mainly used for optimizing surgical conditions, as well as assisting with mechanical ventilation and tracheal intubation. And the mechanism by which these drugs function is not very well known. They know that it mimics the action of acetylcholine, but there hasn't really been much study into how other ways we can, facilitate nicotinic acetylcholine receptor channel closing. So that's part of what I'm looking into today. We're looking more in depth into the structure of the nicotinic acetylcholine receptor itself. In this study, I'm using the Tetraneurus californica nicotinic acetylcholine receptor, and it's made up of 5 different subunits, which I've shown here. So there's 2 alpha subunits, which are shown here in pink, the gamma subunit, as well as beta and delta. And endogenously in your body, acetylcholine will bind in between this alpha gamma subunit, as well as the alpha delta subunit binds in between these 2. Something that's important to note is that humans use almost the same subunit composition, or they use the exact same subunit composition up until 2 weeks post-birth. And then at that point, we then switch to, instead of using the gamma subunit, to the epsilon subunit. So that's something to know as part of the limitations of this study, is that this isn't a direct one-to-one comparison of how this toxin will affect humans. But since there's only one subunit that's different, in the nicotinic acetylcholine receptor, as well as the human epsilon subunit sharing 55.5% identity with the Tetraneurus californica subunit, can still give us a lot of insight into how this toxin could affect binding human muscle type nicotinic acetylcholine receptors. And so looking at just preliminary alpha fold binding, these were made using the sequences of Tetraneurus californica. So this has the 2 alphas, gamma, delta, and beta subunits. And here circled is OSC1 binding. And what you can see, I'm comparing this to other known alpha neurotoxin whose structure has already been solved. And what you can see is that both of these alpha neurotoxins are binding at the alpha gamma and alpha delta sites. Although something to note is that OSC1 is notably lower down here compared to the alpha bungarotoxin. Something important to also note is that the PLDDT value for OSC1 is much lower than for alpha bungarotoxin. You might not really see that well, but here's a key looking at PLDDT values, which stands for predicted local distance difference test, which is a way of scoring each amino acid, and AlphaFold is giving you its approximation for how close to the amino acid's true position is. So as you can see here, since most of OSC1 here is in yellow, there's lower confidence than for alpha bungarotoxin that it binds here. So then I also use AlphaFold to look at how OSC1 binds to potassium channels. These potassium channels, the sequences are coming from mus musculus, or mice, since that's what was originally looked at, that this toxin can bind mice potassium channels. And the KV stands for potassium channel, and there's many different types. So looking at KV 1.1, 1.2, and 3.1, you can see that OSC1 is binding extracellularly. But interesting to note is that KV 1.3, where OSC1 has its highest binding affinity, or it binds tightest to this subtype of potassium channels, you can see it's actually binding inside of the pore where potassium ions usually flow through, which is kind of interesting. And I'll come back to that a little bit later. Now, to give you guys the main goals of this research, first is to model how OSC1 binds to these nicotinic acetylcholine receptors using cryo-EM, and then determining the differences between how OSC1 binds and a similar alpha neurotoxin, alpha bungarotoxin, and comparing their binding to this receptor. Then lastly, going to be interpreting the significance of this binding interaction. So moving on to how I actually modeled this binding interaction. First, talking about how the peptide itself was synthesized. It's a 38 amino acid peptide and it has 3 disulfide bonds. So it was synthesized using solid phase FMOC protected peptide synthesis, and then was redox folded. After that, it was purified using HPLC. The spectra shown here, showing that it's very pure. Then we also use mass spectrometry to double check the mass of our produced peptide. What you can see here, the experimentally determined mass from mass spec roughly aligns with the calculated or expected mass of roughly 4,204 grams per mole. So we're pretty confident that we synthesized the peptide correctly. So then from there, we also need to purify the receptor itself. So this was purified from the electroplaque tissue of Tetranurus californica, and this was done using size exclusion chromatography. Then once this receptor was purified, it then has to be reconstituted with MSP1, E3, D1 nanodiscs, following the methods laid out in Raman et al. So what this nanodisc is, here I'm showing the receptor itself and looking at a map of the hydrophobicity or the hydrophilic nature of these residues. And so this blue represents, these are amino acids which like interacting with water, whereas these yellowish colors here don't enjoy interacting with water so much. So it's important that we add some lipids which are going to be held together using this MSP protein to more naturally mimic the actual nature of these receptors, where this part is going to be embedded in the lipid bilayer. So we need to add kind of a pseudo lipid bilayer just so that the receptor and this or this transmembrane portion is happy and not interacting with water like it normally doesn't do. So now that I have pure OSC1 and the receptor, now we can move on to cryo-EM. So just a very general overview, we load the sample onto a grid. So we use 120 micromolar of the toxin and then 15 micromolar of the receptor. Then just loaded onto this grid and then plunged and frozen in liquid ethane at negative 170 Celsius. Then once we have this grid ready, we can then image it using a cryo-EM microscope. Then we can go through image processing and then model building, where we take the experimentally determined EM density map and insert models into it to try and understand or model how this toxin is actually binding. So a little more about cryo-EM. This is a structural method used to determine the structures of macromolecular complexes. And depending on the target protein, cryo-EM is generally easier to produce samples than X-ray crystallography. And just talking about some of like how cryo-EM actually works, you have this electron detector on the bottom, and you have an electron beam which is shooting down, and it's going to shoot down through the vitrified ice. And if there's nothing in between the top and bottom layer of the ice, the electron is just going to simply go through. But if there happens to be some sort of complex in the way, then these electrons are going to get scattered, and you're going to see that scattering on the detector back here. And from this scattering, you can then reconstruct an image, a 2D image of what this complex looks like. from these 2D images, it's important that we get a bunch of 2D images from a variety of different angles. So this is kind of being shown here. It's important to get images from many different angles since this receptor isn't uniform and it's different along its different subunits. So you need to see all of its different orientations. So then you can take these 2D images and reconstruct a 3D image. And so using the Krios electron microscope, we collected roughly 4,600 exposures that came out to a total starting amount of roughly 7 million particles. So then going into the data processing, once we have all the data collected, the next step is to then remove quote unquote trash data. And so initially we start with these 4,600 exposures, and we're initially just looking for particles that match the size or expected size of the receptor. So roughly 160 angstroms by the 60 angstrom diameter. And in this case, you're going to find a bunch of trash particles in here. So then we do 2D classification, which separates different structures which are in similar orientations. And you can use these 2D classes to further look for better and better quality particles, which you can then eventually use to reconstruct a 3D image. So from those 7 million particles, we went down to roughly 100,000 to get the structure I'll show you on the next slide. Something else that's important to note is that you require a high enough resolution to provide any meaningful results about the binding interaction. So here I'm showing kind of an example of this. I'm shooting for somewhere below a 4 angstrom resolution because at this point you can start seeing definition of individual amino acids, which is necessary if you're trying to determine which amino acids are stabilizing that binding interaction. So this is the initial EM density map that we created, and this was created with roughly 100,000 particles, and it has a 2.84 angstrom resolution. And here are 3 images just showing the local resolution of this structure. You can see on this key here, the 2.5 angstrom shown in dark blue is very highly resolved regions, whereas the farther you get to red, it's less well resolved. So what you can see from just this this EM map of the receptor itself, there's a lot of deep blue around here. There's the farther out you get, it gets maybe a little bit less resolved, but it's important that we have this core structure of the acetylcholine receptor having high resolution, since that then gives us confidence when we're inserting our model to be able to actually fit our model inside of the experimentally determined density map. And so now that we have a good starting point, we can then start looking for the binding site of OSC1. And so as I mentioned, we're going to be inserting the model of the nicotinic acetylcholine receptor of Tetraneurus californica in apo conformation, basically meaning there's no ligand bound. What you can see, like in here, you can see the model kind of poking out of the EM density map. So we're fitting the structure inside of the determined density map. And then what we can do is we can then look at lower density threshold structures. And so these are structures that are more mobile. So compared to the inner structure of the receptor itself, these residues aren't going to move that much. We're going to have high confidence in where they are. But some structures around the outside, such as sugars, which normally are present on the receptor themselves, since they do move around a lot, it's kind of harder to get better resolution of them. But when we're looking at this lower density EM map, what we can see is that there are densities that do correspond to endogenous sugars, and these are circled in red here. So these are all structures where sugars interact with the receptor. And then to find where OSC1 is binding, we need to find sites where sugars don't normally or aren't expected to be present there, as well as this density not corresponding to the receptor itself. And so from our work, we found that this density circled in green here most likely represents where OSC1 is binding. So to then further refine this EM density map, we used a focus mask, which then tailors the 3D classification as well as refinement on the extracellular domain. So this EM map was created with roughly half of the map used previously, around 50,000 particles. And here I'm showing the local resolution map as well with the same key on the last slide. And what we can see is that OSC1 likely is binding the alpha gamma subunit, as well as when we're looking at this structure here, which is looking down the middle, it's kind of cutting this in half. But you can see there's also a density present inside of the pore of the receptor itself, which is pretty surprising. And something to note also is that AlphaFold predicted that OSC1 would bind the alpha delta site, which this would roughly equate to that alpha delta site, although instead of this being a density for OSC1, most likely a density for the sugar that's associated with the receptor. So we only expect OSC1 to be binding at the alpha gamma site as well as potentially inside the pore as well. So then I continued model building and further refinement for the alpha gamma site. And so this is showing the nicotinic acetylcholine receptor as well as OSC1 that has been fit into the revised Coulomb potential map. The map isn't shown here. I'm just showing the models here. And these were further refined using real space refinement in Phoenix. And then the Ramachandran and Rotimer outliers were then corrected in Coot. And just something I'll mention while we're here. So you can see this pink structure here is OSC1. And then here we have the alpha subunits. This is gamma. So here's the alpha gamma binding site. And something to know is that OSC1 is primarily interacting with this gamma subunit here. So before I actually start talking about how OSC1 interacts with the receptor, I'm going to address the disulfide connectivity of OSC1. So the disulfide connectivity wasn't controlled during redox folding. So we were shown here is the expected disulfide folding pattern of OSC1. And here's just showing OSC1 from two different angles, highlighting the disulfide bonds. What we can see is that cysteine 18 binds to cysteine 35 as expected, but then we have this clump where we have cysteines that it seems as though they're all kind of binding each other. And we think that this differs from the determined NMR structure of OSC1 compared to our fitting of OSC1 into the cryo-EM density map. So this indicates that we need to further refine our model, but this doesn't necessarily indicate that OSC1 wasn't synthesized properly and seems to be more of an issue with the refinement. And something else to note is that these intermolecular interactions likely won't radically alter the structure and binding of OSC1, so you can still have decent bit of confidence for binding results. So here we're looking at, I'm just zooming into one point in this figure here, which is a lig plot diagram, which is showing the interactions which are present in between the toxin itself and the receptor. So here in parentheses, K is indicating that this is the OSC1 toxin. And in parentheses E, this is going to be residues that are on the gamma subunit of the receptor. So these red fan looking things are hydrophobic interactions. So we can see, for example, here, tyrosine 111 and leucine 77 form hydrophobic interactions with the toxin, as well as here we have aspartic acid 76, which is forming a salt bridge with lysine 7. So looking at the main interactions that are keeping OSC1 bound to the nicotinic acetylcholine receptor, as I mentioned, here we have this salt bridge, which we looked at on the previous page. as well as there's three hydrogen bonds. One of them is on the alpha subunit, which is here in red. And then this is the gamma subunit, like a pinkish color. Well, we can see there's three different hydrogen bonds being formed. Two of them are on the gamma subunit. Then there's also hydrophobic interactions. Something to note that I mentioned previously is you can see most of these interactions are on the gamma subunit and reinforces that OSC1 primarily binds to the gamma subunit and only loosely interacts with the alpha subunit. And now, since I have a preliminary understanding of how OSC1 binds the alpha gamma subunit, I can then compare this to how alpha bungaro toxin binds to see if there's any key differences. And what we can see when we compare these two is that here this is showing alpha bungaro toxin is this orange structure, and then these blue residues here are showing the residues on the gamma chain which interact, and these are on the alpha chain which interact with alpha bungaro toxin. And something that's similar between these two is that both these alpha neurotoxins interact using cysteine 192 and cysteine 193 on the alpha chain. Although after this, there isn't much overlap. Also, on the gamma subunit, OSC1 doesn't use any of the same residues that alpha bungaro toxin uses to bind. And something I'd like to point out is here is this tyrosine 198, which is on the alpha subunit. And this is a key residue since tyrosine 198 forms a pi cation interaction with quaternary amines. So this is residue which stabilizes acetylcholine binding, and is likely also what neuromuscular blocking drugs use to stabilize binding to the nicotinic acetylcholine receptor. So it's kind of surprising that we don't see this in OSC1 as well. And yeah, as I mentioned earlier, OSC1 has a lot more interactions on the gamma chain than it does alpha. So then lastly, looking into the significance of these results and what to do going forward, I'm going to talk a little bit more about the gamma to epsilon postnatal transition that happens in humans. And seeing as OSC1 relies on the gamma subunit for binding, this could be a tool that we can use to study the differences between the gamma and epsilon subunits. So here I'm showing nicotinic acetylcholine receptor. Here we have the alpha subunit, and then here up on top, this is going to be the gamma subunit and the epsilon. So you can look at the differences between these two subunits and see if it's feasible for OSC1 to also bind the epsilon subunit, knowing what we know about how it interacts with gamma. What we can see here is aspartic acid up here gets turned into a glycine, which I have here in red. This would likely lead to the interaction no longer being conserved. You wouldn't have hydrogen bonding anymore at this site. But then looking at the other two residues, which are, I think this one is used to make the salt bridge, and this is to form another hydrogen bond. So you can see 3 of the 4 primary binding interactions between OSC1 and the receptor are likely to be conserved when you switch from the gamma to the epsilon subunit. So it could maybe be used to study this difference in subunit composition. As well, something interesting to note about the gamma subunit is that it binds acetylcholine with roughly 35 to 40 times greater affinity than the epsilon or delta site. This results in when you have a nicotinic acetylcholine receptor with the gamma subunit, it's going to be open for 3 times as long. And it isn't really clear why this transition happens yet, although preliminary work is suggesting that this switch is initially used for patterning and innervation during development, and then that switch happens after you're developed and out of the womb. Although it still isn't fully known as to why it happens. Now also talking about the OSC1 toxin and how it maybe has an evolutionary advantage. So as was seen, or I mentioned earlier in the talk, this is the KV3 or KV 1.3 potassium channel, which predicted that OSC1 is binding inside of the pore itself. And here we have the local resolution shot of a refined map, which is showing that OSC1 likely also binds inside the pore. And this could tell us maybe how Orthochirus scrobiculosis has an evolutionary advantage that is able to bind multiple different sites which cause flaccid paralysis. And this could also maybe give us some insight into how the toxin actually originally evolved. All right, now looking at the future directions, loosely from most urgent to least urgent, I think the first issue to address is the disulfide connectivity I mentioned earlier in the talk for OSC1. Um, yeah, this would just entail further refinement just to get a better fit into the EM density. And then the next step would be to do the model building process for the density that's located inside the pore, since in this presentation here, we only had enough time to focus on the alpha gamma subunit, so that's also need to be done. Then after that, it would also be important to determine the binding affinity of OSC1 to the nicotinic acetylcholine receptor, since currently only the IC50 value is known, which um, it'd be better if we had a KD value. But then also doing a 2 electrode voltage clamp electrophysiology assay where we're using tissue and actually measuring the change in membrane potential when OSC1 binds to have a more physiologically relevant, I guess, understanding of how OSC1 acts. And then lastly, I would say is modifying key residues on OSC1 that stabilize this OSC1 nicotinic acetylcholine binding interaction to then have certainty that the residues we think are important are actually important in promoting the binding interaction. So now I have my acknowledgements. I just wanted to thank Dr. Stoll. As he mentioned, we've been working together since I was in high school. And I appreciate being able to learn about structural biology all this time as well. I want to thank entirety of Stoll Lab, as well as particularly Lottie Stewart, who mentored me through most of the EM process. And then I'd also like to thank Dr. Day for all of her help inside and out of the lab, as well as Dr. Riley for being my honors chair on my committee. Yeah, that's all.